The most significant map ever created is the human genome. 20 years ago today, the first human genome sequence was published. It revolutionized science and improved our understanding of the course of evolution. What's the next step for us? Today is the 20th anniversary of the Human Genome Project's, HGP, publication of the first sequence of the human genome. When the first draft of the human genome sequence was presented at the White House in June 2000, then U.S. President Bill Clinton remarked, "This is the most important, most wondrous map ever produced by humankind." 3 billion DNA units made up the DNA map, which was effectively a human being's blueprint. The HGP was a significant technological and scientific accomplishment, even though the sequence was only partially complete and was put together using DNA fragments from numerous individuals. Genome mapping held out a lot of potential. We would learn about our ancestors' pasts. We would learn about the role that genes play in disease. To determine whether we may have diseases in the future, we would read our genetic sequences like coffee grains in a cup. However, by that time, we would have created the necessary treatments for those illnesses. Some of these predictions came true, while others were just wild speculation. However, the advent of genomic technology in 2003 marked the start of a revolution in the biological sciences, revolutionizing our understanding of human disease, our evolutionary past, and what it is to be human. The revolution in medical genetics. The biological revolution has been driven by genomic medicine. Since 2003, scientists have sequenced the genomes of over 1 million individuals to determine how genetic variation among individuals impacts disease risk. David Curtis, a professor of medical genetics at University College London in the UK, remarked that it is remarkable that you can obtain the complete genome sequence of a sick kid and discover the cause of their illness within days. For Curtis, the greatest advantage of the HGP is the routine application of genetic technology in clinical practice. We have discovered the first gene for schizophrenia in the previous few years of extensive sequencing investigations. We now have 10. Your risk of developing schizophrenia is significantly increased if these genes are disrupted, Curtis told DW. And we have found a connection between genes and other disorders besides schizophrenia. Researchers have discovered faulty genes that can lead to anything from diabetes to cancer. The ability to develop novel treatments by researching genetic abnormalities in the lab represents the next major advantage. Gene-based medicines have previously shown some degree of success, from employing CRISPR-Cas9 gene editing techniques to treat sickle cell disease to reprogramming immune cells to fight cancer. The field of genetic therapy has enormous potential, according to Curtis. Genetics is extremely useful in the investigation of disorders. Health genetics limitations. However, the recent development has also highlighted the limitations of genetic medicine. The reality remains that few inherited disorders are brought on by a mutation in a single gene, and genes are only one determinant in disease outcome. Only around 1% of persons who have schizophrenia, according to Curtis, have a mutation in one of the 10 discovered genes. More than 250 genetic risk factors for schizophrenia exist in total. A serious problem for medicine is the vast quantity of risk variables and the fact that we don't know how they affect the disease. Some people have a mutation and don't have schizophrenia or have a completely other illness. Since genome sequencing, the entire connection between genetic mutation and disease has grown more tenuous, according to Curtis. Granddad was a Neanderthal, among other stories about human evolution. In evolutionary biology, genome sequencing has been genuinely revolutionary. We may situate our species within that broader evolutionary context by comparing the human genome sequence to that of our close relatives. Anders Bergstrom, an evolutionary scientist at the University of East Anglia in the UK, told DW that it has revealed to us that human history was considerably more active than anyone could have imagined. Our ancestors interbred with other hominins like Neanderthals and Denisovans, according to research into the DNA sequences of ancient and modern humans. We can now trace the genetic ancestry of long-lost people like the Minoans in contemporary Cretans, as well as get insights into the lives of Neanderthal tribes in Central Europe, revealing information about their population decrease. We are learning how our species spread across the globe, introduced agricultural practices to far-off areas, and gained disease resistance by examining ancient DNA and the tiny traces of forebears inside of us. A human is what? The biggest questions surrounding genetics remain unanswered. We don't really know what most of the other 99% of the genome does because just roughly 1% of it codes for proteins. What makes us human is the biggest question that, in Bergstrom's opinion, genetics can answer. Almost all of the genetics progress that has been made in the last 20 years is about explaining differences between humans, but it doesn't tell us about what's universal among us, he added. Genome sequencing has altered how we see ourselves, but mostly by providing evidence to support individualism. According to the theory, since everyone has a unique genetic code, even identical twins don't have exactly the same code. By comparing all the minute genetic differences between individuals, we can learn more about what makes each of us unique. This is frequently fruitful. Because there is more genetic diversity inside racial groups than between them, genome sequencing has demonstrated, for instance, that race is a social construct rather than a biological reality. However, if we are always seeking out differences, we lose sight of what makes the entire human race unique from the other 8 billion ape-like species that share the same planet. Consider chimpanzees. We are aware that we share 96% of their genetic makeup, but we are unsure how the 4% difference distinguishes us from our lice grooming relatives as being more human. It's a fundamental question, remarked Bergstrom. What is it about us that distinguishes us from other species? The expansion of genomic diversity. There were flaws in the advancements made over the previous 20 years. There have been more than 1 million sequenced genomes to date, however 95.2% of the data used in genetics studies comes from European genomes. It did not at all reflect the diversity of humankind. We won't be able to provide everyone with individualized treatment of the same caliber if our science is prejudiced against people of a specific background, according to Bergstrom. The world is evolving. In Nigeria, 100,000 genomes are intended to be sequenced as part of the Nigerian 100K Genome Project. Similar initiatives are being made in other parts of Africa, Asia, and South America. It is hoped that global adoption of more widespread, less expensive genome sequencing technology will be advantageous for all of humanity.